Great. So there is one thing good about this internet meeting, I would say Zoom meeting also, that we can talk from like a long distance and you can attend this lecture from home. But one disadvantage is also that the interaction is minimum here. As like actually, I prefer to have a more interactive session. At least you should take some take home messages after this lecture. I still believe that if you have any question, if you have any doubts, please note it down. Maybe we can discuss it after our this talk. I'm trying to keep it short, and then we'll spend more time on the discussion. And the point here I wanted to make here, and I will go through my presentation, that importance of natural product in drug discovery. That is my main theme area. And idea is natural product has given a lot of molecules in medicine since ages. And unfortunately nowadays people are, or the mainly pharmaceutical industry have not working on the large scale for this natural product driven drug discovery program. They are working in silos, or maybe they are collaborating with the various academic institutions or universities for these kind of discoveries. So I will tell you some of the milestones and of the natural product drug discovery program. What are the challenges associated with, and we will focus on one of the approach which has solved some of the challenges from natural product drug discovery, and that approach is called as de-replication approach. Okay, so title of this talk is Modern Techniques for Identification of Bioactive Compounds and De-replication Approach. So nowadays there is tremendous improvement in the instrumentation area. You might have heard about HPLC, LCMS, NMR, LCNMR, and there are so many I would say advantages or advancements in the instrument part, where the sensitivity has been improved tremendously. And we should take an advantage of that in this field, in this area of research. And that's the target. And let's see, we are working on the similar area. As Dr. Megha has mentioned, that I'm trying to work on the endolycanic fungi, and we will talk about that also quickly, and some of the plant derived molecules as well. All right, so when we talk about milestones of natural products drug discovery, I mean to say that it is having an important role. They have given a lot of molecules, even so many Nobel Prizes is also there. People have backed based on the natural product drug discovery. Okay. However, from 1990, like I said earlier, the companies have scaled back or abandoned their natural product program and focused on the various other technologies such as combinatorial chemistry, HTS approach, high throughput screening approach, where more like human derived, or I would say the molecules which were imagined by or designed by a human mind, where they wanted to focus on. And the primary reason was like it was a biology driven drug discovery program. What I mean by biology driven drug discovery program means, here we understand the disease first. Once we know the disease, we try to understand the target. And then the target also, like enzyme, if it is, then we try to find out the active site of that. Okay. And then we design the molecule which can fit properly into that active site, and it can do the agonist or antagonist effect. So agonist means improve the activity, antagonist means inhibition of the activity. Okay, so the idea is, the people think, and still there are a lot of scientists who still think that we can develop a molecule by our own and we don't need to rely on nature. However, from 1990, there are several reviews in this area and published in the peer-reviewed journal that showing the trend of the novel scaffold coming towards the drug discovery and development has reduced tremendously. What I mean by new scaffold is a new structure that the the nature is doing this kind of chemistry since long time, billions of years ago, and they are producing these molecules in a various way. The biosynthetic pathways are there, and they are, I would say, expert in this as compared to the human being. So there is a tremendous structural diversity associated in a nature, and that is what has been concerned right now. And I want to highlight this because this will give you importance of natural product drug discovery. The point is that if your structural diversity is not that great, then the microbes, for example, if you are trying to find out an antibiotic for particular infection, now the microbes are very smart. They are produce resistance towards the particular drug. And now what if you are aware about, we are getting a lot of generations of the same scaffold, first generation, second generation, third generation. And these are nothing but analog of the first generation or the innovative first scaffold. And what does analog mean? It is like a particular change at a particular site in terms of structure modification. And then what happened if there is not major change in the structure, then the mm, mutation also occurs very quickly in the microbes, and then these microbes become resistant to the newer drug, also newer generation drug as well, quickly. So after some times, it will be very like we need to have a new scaffold, we need to have a new target to find a new drug, so that it will take a good amount of time for the generation of the resistant generation. And that is a major concern right now in the drug discovery or development area. Let's say 10 years down the line, you have to have a new scaffold for this drug discovery business. That's the point I wanted to make. And we have already discussed that from 1990 onwards, the new scaffold and new structure has not been introduced much. There are very less number of molecules, newer molecules, where they're coming into the market from completely different structure. Now, observing this trend, now if you see in 2015, the Nobel Prize was given to Campbell, Omura, and Professor Tu, for the discovery of ivermectins and artemisinin. This decision was very important for natural product research. The discovery of artemisinin, it's a long story. However, it's a natural product molecule. And the Nobel Prize is given because they've also, scientific arena also want to emphasize on the natural product area. And recently in 2019, China has approved a seaweed-based Alzheimer drug, first new medicine with the potential to treat cognitive disease, Alzheimer mainly, it is focusing on. In 17 years, this was like the completely different molecules they have introduced, and that was something which is very good indication now. So again, the I think the interest has been generated, and I just want to make sure that here, though there were less interest from several years now, still around 96%, around 65% of approved antibacterial and anti-cancer agents are derived from natural products. So whole from this slide, I think I'm trying to make it clear that natural product has been always a backbone in the drug discovery area, and one should always focus on this area. Now, though although I've said that it is very important area, for the research. However, there are a lot of challenges, a lot of bottlenecks are there in natural product drug discovery program. And I want to here highlight three major bottlenecks. First of all, before I go to the bottleneck, I want to give you some idea about how this program works. I hope it will help you to understand this. Maybe some of you might be some of you might be aware of this process, but for them it might be revision. For some of you, it is important to understand, because then only you will realize the significance of this process. So generally, drug discovery and development takes more than 12 to 15 years. Okay, and approximate cost, if I'm not exaggerating, let's say consider 1 billion US dollar for one molecule and one drug. And generally, it can be on the both sides, around 10,000 molecules if you start with, which is showing the activity. For example, if you are trying to develop an antibiotic, and you, I'm, I think you have, if you know the disk diffusion method, if this diffusion assay method, where we are growing these microbes or pathogenic bacteria on the agar plate, and then we add a small amount of the drug we make some holes there, wells there rather, and then we add at different concentration of these different drugs, and we see the zone of inhibition here, right? Inhibition is they are trying to kill these microbes, these different compounds or different concentration. We screen this. Now when we observe a good zone of inhibition, we say that okay, this compound has a potential. We also not directly go to the compound level here. In natural product drug discovery, we go first check the extract level, some plant extract or some marine invertebrate extract, or some if it is from the microbes, some microbial fermentation product extract, let's say. Now if you observe that particular zone of inhibition, a good zone of inhibition I would say, compared to your solvent, or I would call it as a blank medium, then you consider as something positive results. And from that result, if you want to end up with that particular molecule as a drug, then it takes minimum 12 to 15 years. If all the processes, if it passes all the steps that we will be discussing now. Okay, so it's a huge process, it's a long process. So uh, that's I said earlier that if it is a biological driven process, then you know the target upfront, that okay, this is the target I want to do work for. I want to find out a molecule which inhibits or which is the agonist for this particular target. Now, this is one of the also important in natural product, or the molecule which is obtained from the biological origin, is that we are really not sure that which is the biological target for this molecule initially, because it is not designed for that particular target, and that's the one of the major concern. That takes a lot of time. Basically, I am trying to say, because you have to screen a lot of target, or you have to nowadays, because of the in silico screening tool, it is becoming better and better nowadays. But still, it is a time-consuming process as compared to the synthetic library, because we are there in that you design the molecule according to the target. So I'm putting it here like a lead identification. Lead identification, I meant to say that, like I said, in terms of antimicrobials, we observe a good zone of inhibition for particular molecule. I am referring to this figure 1 here on the right hand side of this slide, slide number 3. Okay, now once we identify now we screen this molecule, for example, against different biological targets, and then the biological target if we identify it, we need to validate it. What I mean by validation is you have to develop an assay, in vitro assay method, which you can perform in the lab and find out the IC50 value or EC50 value related to this particular target. And this compound can be coming from the various approaches, and that can be anything from the natural product origin to synthetic also sometimes now, if you see the libraries and all that thing. Anyways, so this process, if you come up at the end of this biological target validation process, you kind of, it is after this, all these molecules, either it is coming from natural product or it is coming from let's say synthetic origin, they have to pass through the similar kind of processes. Then now you have a molecule after this which you know that it will bind to the target and give the desired effect. Now, however, you also need to optimize it further for its bioavailability. And if you, that is also another area where you have to modify your structure. Structure optimization is necessary if required, or salt screening is also can be done. So here also you kind of optimize this molecule, which is like giving a good bioactivity, which also gives a good bioavailability. Bioavailability means though there is only one target and one molecule, your molecule will bind to the target very nicely, one to one. But there are multiple targets available here in our system, and the molecule needs to go to the respective target that we are looking for. So we have to make sure that the compound is available for that target, and we have to make sure that it is available in the blood. And so that's the reason for a good amount of time to get the desired effect. Hello, any question? Okay, so so now here at this stage, you kind of come up with the 3 to 6 potential candidate, one backup molecule, one good candidate, which is showing the desired effect, which is giving a good pharmacokinetics, ADME profile, which does not produce a toxic metabolite as well, and which does not give any toxicity here. And then at this stage, now you start working on the preclinical studies. So preclinical studies are not in human, but generally you require a molecule in a good quantity. Generally you produce the molecule in a GMP lab and GLP lab, and you kind of involve now regulators here, which like US FDA or EMEA regulators, where they generally approve this particular molecule. Okay, now here there are some kind of toxicity studies you perform on the animal, and you also perform some efficacy studies, and these studies you document well, and you have to prove that your molecule has minimum or no side effects. You have to prove that the molecule is having a good desired effect, and if there is any competition, you have to show the data against with this competition molecule, and that it something is better or equal, or you have some kind of edge on the existing molecule. Now, once you approve this and you convince to the this your evaluators, this is basically IND application we call it. We investigational new drug application. Here it's almost ready for you, and now this is getting evaluated very nicely with the FDA and all other evaluators, and they approve it for the human trials. And now you have a clinical trials. I'm sure you must have heard about different clinical trials. Phase 1, phase 2, and phase 3 are major 3 clinical trials. Phase 4 is also there, where after approval they are monitoring the side effects or the efficacy or any kind of adverse reaction for this particular molecule. Now in phase 1, generally the molecule has been studied for the on the small healthy volunteers and looking for the adverse effect here. Now phase 2, little bit bigger population, looking for, this is for the patient, and we look for the efficacy and adverse effect. In a phase 3, basically, it is on the bigger population, large population from the various origin. And here, mainly you look for, again, both the efficacy and the efficacy, as well as you are trying to find out here if any side effect or respective origin of the people. Okay, so once the molecule approves or the clear all these stages, then FDA evaluates everything. They recommend few trials, few suggestions, few changes, and then they approve the particular drug. So, like as I told you, it's a very lengthy process, more than 12 to 15 years it takes, and if you start with 10,000 bioactive molecule, we call it as a lead, then you end up with one new drug here. This is general trend. Now, although this is a long process, now if you are working in a natural product area, it is even lengthier process. And what is the thing there is that here in the natural product area, you have a natural product source, any source, let's say a plant source here. Now in the plant source, you kind of get the extract first, then you're trying to find out the bioactive molecule, and then you try to find out the structure of that bioactive molecule, the identification. It is also itself a lengthy process, may take sometimes 2 years if the molecular weight of the molecule is, let's say, 1,500 or maybe more than 1,000. Then it takes a good amount of, I would say, months to do the structural elucidation of that particular molecule, because you need to find out the absolute stereochemistry as well. It is not only the two-dimensional structures you can end up with. So now what happened here in natural product case, when you spend this much time on the particular specimen, and at the end, if you come to know that it is a known compound and somebody else has already reported for this activity, then it is a kind of repetition of the job, repetition of the work. This is one of the most, I would say, problematic situation for the natural product researcher. And another issue is that, let's say you have isolated a compound from 10 kg of plant material, and you end up with isolating, let's say, 5 milligram or something. So for 5 milligram, you need a 10 kg of plant material. So that means the yield is very less, yield is very poor. And it is a big issue, because you cannot keep cutting the plants, you cannot keep cutting the trees for your compound. So the supply issue is a major thing. Then, as I already told you, the third bottleneck in natural product drug discovery is the biological target identification. Because sometimes if it is not the known target, then to find out unknown target is a Herculean task, and it takes a good number of years to find out the target, actual target of the particular molecule. And these are, I would say, the major bottlenecks that that has curtailed down the natural product research in the most of the industry, because the industry doesn't want to spend time in the thing which you are not sure about, and that's the major thing. And now, out of these three problem statement, here I'm going to show you some kind of a positive note for the resolution of the one problem statement, one bottleneck, which is what I said, isolation of the known compound. I will repeat the problem statement here, because maybe it will help everyone to understand what we are talking about. So let's say we have a plant material. You have prepared various extract, various solvent extract, and then you have fractionated that particular extract. So now, for example, in any plant material, let's say you have more than 20 fractions or 50 fractions, for example. Now, out of that 50 fractions, you have got, let's say, 4 fractions which are bioactive, you have found out. And then from that 4 fraction, you have isolated several compounds from that. This procedure takes good number of time, as sometimes 6 months or maybe sometimes more than 2 years also. But most of the time what happened, there are higher chances that you come up across with the already reported molecule, which is being reported by the previously somebody by in other uh, maybe other plant, or maybe the similar plant, or maybe some other species from the same genus. All these things, all these issue or all these things can be possible. But then what you can do, let's say you have already let's say you are screening for the anti-cancer molecule, and now you have observed, you have got a molecule which is already known reported molecule, and its activity is also known, anti-cancer. Then it is of no use of this particular molecule. But you have spent already a year or maybe more on that particular project. So it is a kind of wastage of time, wastage of efforts, and everything. The cost also. Now, to avoid this, we are using a process for the early identification of these known molecules. And this process is called as de-replication process. Now here I have mentioned a bag of tricks. Bag of tricks is nothing but a method, a process where you try to identify this compound before isolation of that particular compound. So you don't spend time in the isolation, characterization individually. So you spend time in the extract or a fraction level itself. So it's the process can save a good amount of time, where from the time frame can move from 6 months to maybe 1 to 2 weeks, and that is the main advantage. And you can see on the right hand side here, these are the number of publication on the de-replication process. And you can see that this was up to 2013 actually, but I didn't find a review mentioning this kind of analysis. But I'm sure the number of papers on de-replication has improved now a lot. And so as you can see there, the bar has been increasing from 2004 to 2013, as the number of publications on this de-replication process has increased, mentioning that the people are following this kind of technique a lot in the natural product research. The thing is, any method, and we will discuss these methods now in the coming slide. Any method you apply, your idea is to find out the that compound, if it is known or if it is unknown. Now, if it is known, you try to avoid working on that particular molecule and you keep it aside. Here I put it in the trash can. Now, if it is unknown, then we have NCE, that is novel chemical entity, to work on. Then it is obviously a good advantage to, though you have to work on the structural elucidation part, but the chances, the probability of having that molecule as a new molecule are very high here. And that is the advantage here. So if you are working on a novel scaffold, it gives you more pleasure to work on. And that is at the end, you get more and more satisfaction saying that, okay, you are end up with a new compound at the end. So this process can be considered as a filter in between two steps. For example, from fractionation to isolation and characterization of the molecule, you can add this as a filter, and it will remove the molecules or remove the fractions, I would say, which are containing the known compound. And that's the aim of this particular process, the method. At least I want to give you this as a take home message today at the end of this lecture, that okay, these type of methods, this type of techniques exist, and they help in natural product area to identify the molecule at an early stage. And now we will discuss how this technique work. Okay. Basically, as I said earlier, that there are so much advancement in the instrumentation part. HPLCs are there, lot of uh, various detectors are there which are attached to the HPLC. GC also a lot of detectors are attached. And so one of the major detector is MS, which is a mass spectrometer. And PDAE also, I think some of you might have used HPLC with UV-Vis detector or a PDA detector. And then there is an instrument called as LCMS, where the HPLC is like that, the same as this PDA detector, but one additional detector is attached, which is a mass spectrometer detector. Mass spectrometer detector is a unique detector. I mean to say that here you will get information about the molecular weight of the particular peak or compound which is eluted at particular time in the chromatography. Now, here the advantage of chromatography is that you are separating the compound in the column, and one by one you are sending it to the detector. And that is the advantage, as you do not have to isolate the compound now, because it is being separated in the chromatographic column. Column means HPLC column itself or the LCMS column. So here how I would read this and then we will discuss about it. And the process is the quick identification of known substance by advanced instrumentation is called as dereplication, which allow researchers to focus their efforts on the discovery of new ones. Different hyphenation technique used in dereplication: LCUV, PDA, LCMS, LCMSMS, which is nothing but the fragmentation, LCNMR, and LCMSMS-NMR. And there are furthermore techniques which include GC, GCMS, GCMSMS as well.
डेकोरेशन में क्या क्या कुर्सी भी इंक्लूडेड है
Okay, could you please put your mic on the mute mode?
गजीबो
Please put your mic on the mute mode. Yeah, thank you. Like I said, there are many methods here you can apply, but request you not to write anything on the screen, please. Okay. So these methods have an advantage that you get some information about the molecule, and that is the beauty of these methods. Where in case of mass spectrometer, you get the molecular weight. There is one more technique here which I have not mentioned, which is LC-HRMS, which is a high resolution mass spectrometer. And in that case, you get a accurate mass information. Accurate mass information is nothing but the mass up to 3 decimal places or up to 4 decimal places. Accurate mass. So, for example, if you say my molecular weight of compound is 371, I would say the accurate mass of this particular compound, monoisotopic mass, is 371.3257. And then with that accuracy, you find out the molecular formula for that particular compound, which is very, very helpful in identifying that particular compound from this analysis. You don't need to really isolate for that. That's the particular advantage. Now, like I said, you have various instruments which can be used for this particular analysis. However, only analysis will not help. You have to have a database here, because you have to compare with something. You have to generate some database first, and then whatever the acquired data is there, that you compare along with this database. And so there are already available databases are there in the literature, and few of them I have mentioned here. These are just for a few for the discussion, but there are many more databases. In fact, you can create your own database by using your own isolated compound. If let's say you have 1,000 or 2,000 compound in your lab, you can create your own database. You can create your own library using LCMS or using LCMS technique. And this is very important because you have to compare with the something, and then only you can identify it. So for example, in the antibase, you have a database of, let's say, around 60,000 compounds are there, which are from the microbial origin and mainly work as antibiotics. Then there is a marine lit database, which is like a molecule isolated from the marine environment, which is there. They also around 40,000 molecules are there in this database. And nowadays, we are getting a mix of these two, which is called as anti-marine, and you have a combined database for these both molecules. Then there is an app alert. There is a dictionary of natural products. Dictionary of natural product contains almost 290,000 compound, and which is from the natural origin. And there is a sci-finder also, which is basically CAS. You have all the chemicals as till date is there. You can find in the sci-finder. Now, in the dictionary of natural product, the advantage I would say is that it is not a. There is a hard copy also, but it is not only a book. It is even you can search that like you are searching in the Google. You can search. I request everyone to please keep your mic on the mute mode. I'm getting some disturbance. Please keep your mic on the mute mode. Thank you. Okay. So, like I said, it is not like a hard copy, hardbound book that if you know, let's say I have a compound with a molecular weight 371. I know that's accurate mass also 371.3251. Now I need to find out if any compound with this information, it is already reported and it is there in the dictionary of natural product or not. If it is a hardbound book, it will take a lot of time to find out. But if you can search it like a search engine, okay, you can have a filter of molecular weight, you put that, and then you filter it. If you have a filter called as accurate mass, you add your accurate mass and you filter accordingly. Now, out of these 290,000 compound, you will end up with you will end up with finding out, let's say, 70 to 80 hits having this molecular weight. Maybe you will have 10 to 20 hits with this accurate mass, maybe 10 hits. Now you know the plant name, the genus and species. You can add as a filter as well here. And then you will then it will show you maybe one or two hit only. And so you can identify it very easily. Next step is very simple. You can compare the one or two molecule which you have observed as a hit. You compare the like retention type information. You compare the UV profile of this particular thing. That can be compared as additional information. And if it is matching with your fraction, which is like in nowadays, if you have a PDA detector, you can get a UV spectrum. You will get a retention type information, which is nothing but give you a structural information, like if it is a polar or non-polar and so on and so forth. And with this information, you can easily identify that this is the same compound or not. Generally, in accurate mass, we find out the mass ppm error. So the observed mass, accurate mass, and the reported accurate mass difference. If it is more than 10 ppm, then you say that it might be different molecule. But if it is less than 10 ppm, generally some if you go more stringently, we say the 5 ppm, less than 5 ppm. It depends on the instrument error also, which instrument you are using, because some instrument has a less accuracy, there the ppm error may be a little bit higher, like a off time of flight and something. But in some cases like Orbitrap machine, you get a very accurate mass information. And in that case, you can go with maybe less than 2 ppm error. And that is the advantage here. And when you say that this compound we have less than 2 ppm error with the reported one, and having all the parameter matching, same plant, same genus, same species, then there are more chances, the probabilities are more than 90% that it's the same molecule. And then if you identify the molecule here, you do not want to isolate again the same molecule if your bioactivity is the same. So as you can see in the right hand side here, you do the fractionation, you then go for the de-replication. Now you screen for the bioactivity, because now here if you know the molecule, it is a known molecule, then you see the reported bioactivity of that particular compound. Now here one thing is there, for example, if the compound is reported as antimicrobial and you have observed anti-diabetic activity for this particular molecule, then don't ignore that. It is also very important, and that is called as NCA. So if it is a novel scaffold, it is called as NCE. If it is a known scaffold, but new activity, it is called as NCA, novel chemical application. So this is also very important. And but here you isolate the known compound, but with a different activity. And so your efforts are not wasted here. People are using this tool also to find out the known with the new activity. If you find no hit in the database, so for example, your molecular weight is completely different, or accurate mass is different, or you are getting a completely different UV profile, then in that case, you isolate that compound, and then you get mostly end up with the NCE, which is new chemical entity. And now I will give you, we have discussed this as these techniques are getting more and more attention, more and more publications are coming out of from this. And I will tell you some of the examples also, which we have used in our lab. And these are some of the publications that I'm showing you in this slide number 8, that they talk about the de-replication approach, and they come up with the new methodology, they come up with the new molecules at the end of this technique. And so that's the reason this is very, very important tool, and I want everyone to be aware about it. Unfortunately, in India, very few scientists are working on this technology, tools, but most of the colleges and labs are working on natural product area. They are isolating compounds, and they are isolating again the known molecules again and again. So this is very important, that one should be aware about this technology, this new trend, and people are following it. It is not only in the drug area, it is also there in the food application as well. This is the example where the people have done the metabolite profiling for the maple derived products using these de-replication approaches. And so they have kind of profiled the metabolites which are present in the different bark, uh, different extract. So very, very important tool. I want you to make sure that today you understand it clearly. If you have any questions, we can definitely discuss on this. Now I'm going to tell you more on the examples and more on the case studies about these techniques. So, as I said, natural resources, it's not only terrestrial plant. We have worked a lot on the terrestrial plant, but now it's time to work also on the marine invertebrate, marine plants, or the marine samples. Uh, it's also a time to work on the microbes. Why? Because in the terrestrial plant now you're working, you are ending up with getting a known scaffold, a known molecule. Now, as I said, this we as a natural product researcher should involve in the research where you kind of give novel molecule to the drug discovery program. And that's why you have to go with a different biodiversity. And in the marine environment, the biodiversity is huge. And so that's the reason this should be explored. Then microbes also need to be explored from the soil as well as from marine environment. Why microbes? Because I told you the supply issue, one of the challenge associated with the natural product research. Yeah. So supply issue is very important because you need a sustainable method. Sustainable method mean to say that now you have identified a molecule, isolate a compound which is having bioactivity, but it is available in a very few milligram level. For the drug development process, you need multiple, like in grams or in kgs of this particular pure compound. From natural product, getting kgs of pure compound is like very difficult thing. And that's the reason the people are moving towards the microbes. And microbes has a tremendous capacity. And tremendous capacity, I mean to say that they can produce a number of scaffold. They can produce a scaffold in a good quantity as well. For example, if you isolate a microbe in the lab and you found that the microbe is producing, let's say, a fungi or a bacteria, for example, actinomycetes, anything, they are producing a molecule, and now you don't have to go back again in nature for the source, because you have that microbes in your lab now. You ferment it at a higher scale, 1,000 liter fermenter, 10,000 liter fermenter, it depends on your requirement, and you process that to get a good yield, to get your molecule of interest at the higher quantity. And so that's the reason the people are now focusing and shifting gears to the towards the microbes to find out a new scaffold, to find out a new compound in natural product area, because you don't have to cut the trees, then you don't have to go to the marine invertebrate, you don't have to go to the marine environment for the collection and to disturb the environment again and again, because it is also a restricted area. So fortunately, I was involved in all these natural sources. My PhD is from. I have done isolated various molecules. I've done my PhD from Florida Atlantic University, and I've worked on this coral, which is you can see here in this diagram. And this we have isolated various new compound using this dereplication scaffold strategy there. And I've worked on this marine, several marine organism. In fact, marine microbes also we worked in the piramal to find out a novel scaffold. Worked on the several plants also, and we will take one case study in the plant today. And in fact, the microbes, when I was working in the industry, mostly in the piramal life sciences, we were focusing on the microbes because, as I said, supply issue was not there. And we have already got one molecule up to IND level from the microbe, and that molecule we have isolated in a kilogram quantity. You can imagine we have optimized the process for fermentation, optimize the process for the extraction and purification to get this at the larger quantity. So this is something which is very important if you want to take a natural product to the next level. And like I said, the automation here played an important role. Automation means the instruments are doing, helping us to do the job very quickly. And here you are saving the time. Here you are saving, you are reducing the error which can come through the human interference. Okay, now this is the picture on the left hand side. It is called as a liquid handler. And if you can see my screen cursor, this is a liquid handler. Basically here, this can be utilized to inject the sample as well as to collect the fractions also. So here you can see that this photograph shows you different chromatograms, different peaks, and that can be collected. The different vials here are different. In 96 well plate, you can collect it and send it to the bioassay, which is this is high throughput screening photograph, where you can screen the 96 well plate or even 256 well plate in one go and find out that which fraction is active and what is the activity. Here you are seeing the UV profile of that particular peak. In fact, some of the cases, mass-based purification techniques, you can see the mass spectrum here at this in this particular forum directly. Very helpful. These kind of trays you can directly keep it into the machine, which is called as gene vac or speed vac, and there it will dry, means you can get rid of the solvent quickly, and so that it saves your time and efforts also. This is very important to speed up the process, because the time is very important, and that's why this kind of technology has helped natural products to again come into the picture in terms of industry as well. And there are several nowadays case studies you can find out where the industry academia collaborations are happening, and academia is trying to find out, academia I mean to say the institutes as well as the universities, they are trying to find out the actives, and they are giving to the industry in the like early discovery phase level, and then the industries, because which are set for the development processes, and they are taking those leads from there and going to the further development of these molecules. This diagram is there, which is my own process which we worked here on this, as we used to have two machines here. One is HPLC attached to the column and PDA detector, and mass spectrometer. But here we used to have a division. We divide the flow. 10% only used to go to the mass spec, and 90% we used to send it for the bioassay test. So why I'm discussing this, because now I'm telling you the workflow, where earlier we used to spend 6 months in that. Now in 6 days, I would say, or 1 or 2 weeks maximum, we can get the data out of it. Now here we have worked on the project where we have screened 700 microbial cultures collected from the different part of India. This was the DBT funded project, and this we use this technology to screen this different microbial project. We have got very good molecules that we have submitted to DBT after at the completion of this project. Now, what we did here, that in one injection, we optimize the injection quantity such a way that we get a good amount of particular fraction in each well. And if the activity is in higher nanomolar and micromolar range, you can see that very clearly in this bioassay. So this bioassay is nothing but, if you can see on the right hand side, this is basically you come in a chromatogram, you put it in the different well, say 0.5 ml, you put it in each well, and this runtime was around 15 to 20 minutes runtime. Depends on the sample to sample basically, but not more than that. And then you try to get the information on each data point in terms of mass analysis. So each point you have a mass spectrum. Now you have a PDA data as well. You have a mass spectrum data. We do not analyze it early. We first take the bioassay data first. So we dry this plate and we send it to the HTS department. They get the activity done. So for example, this is our activity data. And you can see that at this particular time, you can see some of the activity here. Now, at this particular time, you check the chromatogram. If any peak compound is there, then you check the mass of that compound, molecular weight, and UV profile, and look into the database that, okay, any compound with this activity. So here we used to send it for the 4 different activities. We used to send it for anti-diabetes, anti-inflammation, anti-cancer with the 3 different cancer cell lines, and anti-infectives, which is for against some pathogenic bacteria and fungi. Whichever area you get the activity, we used to consider it for the further analysis. Now, by doing this, we could complete one microbial one microbe or one microbial culture analysis within a week. Week also it's a little bit longer, because within 3 days we used to get the bioassay data, and 1 to 2 days for the analysis purpose, and hardly 1 hour I would say. If you consider the method setup and all these things, maybe 2 hours and in 1 hour. Each run maybe 30 minutes, maybe you are repeating it, so 1 hour only for the particular analysis. Now, one more thing we used to do, because the microbes, though they are collected from the different origin, maybe sometimes you end up with isolating the same microbe. And so that's the reason we used to group them. We used to cluster them. So this is called as a chemical fingerprinting based on the what kind of chemicals they are producing. Here we add 3 detectors: ELSD detector, UV detector, and mass detector. So there is an additional detector here, which is a ELSD detector, because some compounds are not UV active. Some compounds may not get detected in the mass spec, but in ELSD, it is most of the compounds you can see it here, and their abundance also, which represents the concentration. And so that's the reason. If you have, for example, 5 cultures which are producing the similar type of the profile, then you consider one of them, and that's what we used to call as a clustering. And in the cluster, we take our grouping, we take one culture from there and we take it further for the further characterization. So here with this process, this is called as the replication process, which is nothing but the screening process. And from here, if we found out that okay, the bioactivity is there, the unknown moiety is present in the fraction, and which is also different in this cluster, then we used to select that fraction for the large scale fractionation. And large scale fractionation is nothing but this. So this is one of the project from the plant based, and here this was the profile we got from UPLC, which is a little bit better version of HPLC, where you can go with a high pressure. That means the column, you can go with a lower particle size. And so that's the reason you get a better separation power. And based on this profile, we did a vacuum liquid chromatography fractionation. This is how then all the fraction would look like. Then we perform the LCMS analysis and the bioactivity and observed that in the 40% fractions, we found some novel compound is present. We further developed the method for the 40% fraction with the HPLC, and then we checked with LCMS as well. And then with the prep HPLC, we collected these peaks here, as we can, I'm showing you as an arrow. And these compounds were then carried out the structural elucidation part, because it has shown that if the LCMS analysis, de-replication analysis, that the novel compound is present in this fraction. And this is after purification, relatively pure compound. You get the molecular weight information, molecular mass information. You get the UV profile also. This is the same compound, which was purity we estimated as greater than 98% pure. And this was the UV profile of this particular compound. Now, when we performed the MS-MS fragmentation in both the positive and negative mode, in our library, we also had a library. In our library, there was no hit. Otherwise, it would have shown a hit here. And so that is why we have considered it as for the further characterization part, which was done based on the NMR data, extensive NMR analysis, proton, carbon, depth. This depth analysis is nothing but which can distinguish between the CH3s to CH2s, CH2s to CH, and then quaternary carbon, we used to get it in the carbon 13. So then the COSY, which is nothing but a 2D NMR technique in which you have a neighboring proton having a cross correlation. For those who are new to NMR, maybe a little bit difficult to digest this, but I just wanted to give you information that these kind of NMR techniques are there for the structural elucidation of unknown molecules. Here you get information about the partial structure. You come to know that this is a proton, which is an environment of this, like say phenyl proton, or it is a proton which is a double bond, or if it is a proton which is near to the heteroatom, and all this the information you get in this NMR technique. Then there is a HSQC and HMBC, which is a heteronuclear correlation, also like carbon and proton correlation. You get the information using this NMR technique. We also perform LCMS analysis, GCMS for the mass data, then HRMS, then UV characters. For UV, we have done the UPLC and UPLCMS and understanding. So with this, all this structure elucidation, we then end up with one new scaffold, with new structure, which was then screened against with the help of structure and checked in the database if this is already been reported by anybody anywhere in the world, and it was not reported earlier. And so this was earlier we tried to put it in the patent because the activity was there. But then later on we decided to go for the publication, and so it will be getting published soon. So this is why I'm showing this example, because we end up with getting novel structure by applying the dereplication strategy. And that's the message I wanted to give you, that still there are a lot of things are there, unknown molecules are there in the nature, but we need to explore them, we need to focus on them, we need to change the strategy to find these molecules from the nature. And that's our job and the new generation people. We do not want to follow the same path that our previous researchers have followed. You follow the different path, you will end up with the different molecules. And that's how the idea should be. That's how the effort should be, to find out novel molecules from the nature. So I am now quickly sharing some of the data which we have worked on. And this is from the, like I said, from the coral. We use in-house library for that. And we have isolated, though from this coral already 28 molecules were reported previously, and I could isolate 9 new compounds. In fact, one completely new series of molecules that we have isolated from this, because we have used this kind of strategy, and that was the strength of this particular project. We published it in Journal of Natural Products, these molecules. Now, here we are working on endolichenic fungi. Like I told you earlier also, the microbes are, I'm not taking much time now, maybe give me another 15 minutes. We'll try to complete this quickly. Now, as I said, the microbes has tremendous capacity. Microbes are not staying independently. They are residing inside the plant. They are residing inside the other organisms also, right? We also have a lot of microorganisms in our sphere, good bacteria, bad bacteria. And so they help us for various actions. Same in the plant as well. There are a lot of microbes, a lot of bacteria, a lot of fungi. They are inside the plant. And these are called as endophytic fungi or endophytic bacteria. Now, endophytes, they are called. Now, we wanted to work on, because I told you already, the different biodiversity, different chemical diversity you'll end up with. Now, our idea was to work on the endolichenic fungi. So lichen is a fungus. It is a colony forming fungus. And we wanted to work on the fungus, lichen, which grown on the mangrove plant. You know, this here you can see the Avicennia marina. This picture is from Khambat region, Gulf of Khambat, where we went for the collection. And now the lichen grows on this environment, which is humid, and there is a lot of salinity there. Very drastic environment. It is difficult for any biological species to survive. And so if they are surviving in a very harsh environment. They are producing different chemicals. That was the hypothesis. So we have got the lichen from there. Here we are also collaborating this with our with one of the Sri Lankan university, which is University of Kelania. Professor Priyani Peranagama is our collaborator there. So in this project, we are working on the not the lichen as it is, because again we cannot go back into the nature and collect the lichen in the large quantity. We want something which can solve the supply issue. And so we are working on we are isolating fungus from this lichen, which is an endolichenic fungus, which is the fungus which is residing in the thalli of lichen, inside lichen. And that fungus we are growing in the plate and in the now the fermentation also. And we are trying to find out what the different molecules they are producing. Now, we have identified already some compounds produced by these molecules, and some of the compounds are there in the progress right now, but we propose that this might be NCEs. Unfortunately, the quantity is very less, and so that's the reason we are trying to further ferment it and then get the large quantity. And then, because we are working in the now academic institute here in NIPER, and in that institute fermentation, large bigger fermenters are not available, and so we are working on a smaller flask fermentation, and we are getting this compound in the and further structural elucidation. One example I want to give you, like how we do and how it works. Generally I give that as an assignment, but here in the online forum it is difficult. But just I wanted to give you an example, because then you will realize the importance of high resolution mass spectrometry. This is the example, very simple example. If you focus, I think you will learn it very quickly. A molecule with a mass of 44 could be either C3H8, you calculate it. Atomic weight of carbon is 12, hydrogen is 1. So 12 into 3 plus 8. So 44. Same for C2H4O, same for CO2, same for CN2H4. You will end up with a 44. So when you get a mass molecular weight 44, it can be one of anything. Let's say this one of these 4. But you have only one compound. Now, when you go for the exact mass or accurate mass, you get the information, let's say 44.029. Now with this, now when you use monoisotopic mass of these atoms, like for carbon, it is 12.0000. For hydrogen, it is 1.007825. For oxygen, it is 15.9943, I think. And for nitrogen, also it is, you get the accurate mass there. When you add this, you get these values: 44.0626, 44.02620, 43.9898, and 44.03740. Now you know the closest one here, which is this one. And that's the reason you can quickly identify here that our molecule is, the molecular formula is this C2H4O, and hence you can come quickly to your structure, which is very important. This is another example that you can find out. Nominal mass is same, but the precise mass or the accurate mass, which can be distinguished by the HRMS value, are different. And hence, this has a powerful information, and you can quickly come up with the structure if you have knowledge of this. Because there are a lot of databases, as I said, they shown you earlier, they use this high resolution mass spec data, and you can screen very quickly. We have here at NIPER Dictionary of Natural Products, which has all this information, and we have like around 290,000 compound in that database. So we do not spend time in isolating the same molecule again and again, which is there in the database. We try to focus on the new thing. These are some of the in-house projects that we worked on, and still we are working on some of them to find out a new compound from there. And today I'm going to share one case study, which is the last case study for my talk, and which will take not more than 10 minutes. Then we will take question and answer. Mm, uh, Bahonia viragata, which is a well-known plant known as kachanar. Lot of pharmacological activities. There are 19 compounds reported in DNP, anti-cancer, anti-diabetic, hepatoprotective activity. We have taken some plant material, around 350 grams initially, and performed some extraction, and then the anti-cancer assay. Now we have also generated a fingerprint also, which is the HPLC fingerprint, to see that what kind of compounds are there in this particular fraction. Now we did LCMS based dereplication, which is HRMS based, and we identified the hit along with the ppm mass ppm errors. We have observed some hits as which are shown in the structural form here, but some of the we have got peaks which are showing the no hits. And so that was the interesting part, because we wanted to work on the compounds which are novel and for which no structure has been reported. And so we went ahead, isolated, and this is one of my student worked on this, master student on this particular project. She has done the fractionation with open column chromatography, glass column, and then all the time we use some HPLC analysis also, or the LCMS analysis for the shortlisting of this compound, because we kind of analyze our extract earlier, and we knew that which type of molecules we wanted to work on. So this was the example here. As you can see there, these molecules which is having the absorbance at three different wavelengths, 254, 365 and 620, which were there, and which has shown also some moderate IC50 value against MCF7 cell line, which is the anti-cancer cell line for the breast cancer. Now this we wanted to purify these molecules, and so we have developed an HPLC method, then the preparative HPLC method, and then we have isolated these molecules, one of the molecule actually, because we started with a very small quantity of the plant material. Couldn't get a good amount of this particular compound, but nevertheless we have identified that the compound is bioactive, as we compared the compound activity against one of the standard, and which is very good, 0.117. So lower microgram means higher nanogram level activity for this compound. Then we screen with again another cell lines as well, and we found a better activity with IMR 32 cell line in the pure compound. And that is the advantage now, that now right now we do not know the structure of this molecule at this stage. We are now working on to isolate this molecule in the large quantity, because of the a very small quantity we have isolated earlier. But we kind of sure that we will be end up with getting the new compound with this, because we have not got any hit for this compound, and we know the scaffold also. We have done the NMR of this because of the limited quantity we get the NMR. We know that the scaffold, but the exact structure, the three-dimensional structure, the chirality, everything, if you want to establish, you need a little bit better quantity. And so my another student is now working on this to get this molecule in a large quantity to get the exact structure. The point here again, I wanted to make sure that we applied the dereplication technique on the known plant, which was been studied earlier, but we could get the novel compound, the NCE here. And like I said, that was my end of my presentation. And we will discuss more on the questions that you have. I just, before concluding, I want to acknowledge my student, because they are working on the various projects right now. Some of the PhD students, some master students already passed out. This Ashwini Ghogre's project data I have shown today in my slides. So I want to thank her for that, and some of the Aishwarya and all, they have helped in the BIIS-7. I also would like to thank our director, Professor Kiran Kalia from NIPER Ahmedabad, for the support on the conducting various projects for the funding, as well as, and also thank Department of Science and Technology, DST, for funding this endolycanic project. With this, I would like to thank you all for your patience listening to me, and I would be definitely very happy to answer any question if you have. Thank you.
Hello, sir.
Yes.
Sir, please let me know seaweed-based Alzheimer's drug, sir, which approved by the
Yes.
China in 2019, sir.
Yes. So it is basically a polysaccharide type molecule. Okay. And so that is not maybe currently it is not the, I would say, the enriched fraction they have came up with, and that's why it is there in the right now. It is still a lot of questions also there from US and Europe for that particular molecule, but it is a polysaccharide organolactin, some name they have given. So it is a polysaccharide type molecule. Okay.
Okay, sir. Sir, second question. Sir, why the database is important, sir? So once again, sir, let me know, sir. And the fraction in extract for new drug delivery, sir.
So to answer your first question, why database is important, because from these instrumentation you will get only the information from your sample, right? But to confirm the structure, you have to have some kind of comparison with some known thing, because we are working on the known identification of known compound, right? So for that, it is important to compare with something, some authenticate, either a spectral database or a particular compound. So you can create your in-house library if you have the compound. Otherwise, you can trust on these known databases, which are very well, I would say, certified databases, and they have prepared it diligently so that you can trust on that also.
And sir, my last question. Sir,
Yes.
is the calibration is required for all analysis equipment as like high resolution mass spectrum?
Yes, very good question. Yeah, calibration is something which is very important for the particular instrument. So in terms of, I would say, in accuracy, mass accuracy. So if you have a tough time of flight instrument, then generally we do every day calibration. So for this machine, because the temperature also create some kind of disturbance, and this is very important. And the calibration is nothing but we call it in the mass spec is a tuning. And in the morning, generally before we start the experiment, we perform the tuning of the mass spectrometer, and with the known compound, with the known which generally comes along with the instrument, we kind of tune the instrument, and then we apply for our unknown sample. Yes, so it is very important calibration. Even for the unknown quantitative analysis also, the calibration is very important. Yeah.
Okay. Thank you so much, sir.
You're welcome.
Sir, good
Yes.
afternoon, sir. I had a question.
Yes.
So the databases that you mentioned, that the antibiotic database and the marine database and others as well,
Correct.
do they have the 3D structure as well of the natural compounds?
Yes, yes. They have the structure, they have bioactivity, they have like source information, like which plant or what is the marine invertebrate that they have come up with. They have also detailed about like what is the UV profile of that, and they have different sections in that. All this data has been filled properly. Yeah.
Because at sometimes PubChem is not enough to search for the natural compounds. That's why I was asking.
Right. No, it is not definitely. You should focus. SciFinder is the updated one, the most updated one. Dictionary of Natural Product is kind of getting updated once in a 6 month, I would say. Okay, so they generally Dictionary of Natural Product, they have very stringent screening process, and so that's the reason any published data they analyze by themselves first and then they put it in their database. So that's the reason it takes time to get that molecule into their database. You understand what I'm trying to say? Suppose you are published today something, one new compound, okay? So that data, this Dictionary of Natural Product people will analyze by themselves first. And if they satisfy with the requirement, then they will put it in their database. Otherwise, they will ask author some more data. And it takes time sometimes to get this molecule in the Dictionary of Natural Product.
Okay, sir. Thank you very much.
Sure. Welcome.
Good afternoon, sir.
Yes, good
Sir, I am Satyen Singh from Lucknow. Sir, I have one question to ask. Sir, how we can enhance the bioavailability of any natural compound which we extract from medicinal plants?
Very good. So bioavailability, the main thing, actually two approaches people are using to improve the bioavailability, because this is one of the major concern. One is a salt screening. We people are trying to make a various salt of these particular molecules, and if you can make a salt, if you can improve the bioavailability of this particular molecules. That is one of the approach. But if it is not successful, then there are formulation approaches as well, where you can go with a site specific formulations, like some of them are the nano formulation also people are working on right now. One more thing is that if possible, because sometimes in natural product, if you try to derivatize them or if you try to play with them, you may lose the activity also. But if possible, if you are not losing the activity, if the molecule structure is small and you can play with it, then SAR also can be planned, which is a structure activity relationship. In this case, the activity may not be the efficacy. Here, the activity is kind of bioavailability. So we change the structure and see that if you are retaining the activity, at the same time, you are trying to improve the bioavailability. We have tried to work on this one of the molecule, which is the concordamycin molecule, where the activity was very good, but the compound was not having. In fact, we were trying to use it for rheumatoid arthritis, and the site of injection, we used to get the necrosis. So the molecule used to precipitate at that particular site where the person used to inject it at that. So what we did was we tried initially salt screening and this different formulation, but it didn't work out. So we finally decided to go for the structure modification. We replaced ketone with oxime and then prepared the I think the format acetate or some salt of it, and then that molecule then we tried with this for the activity, and then it worked well, and we kind of overcome the problem of the necrosis there, where the site of injection they used to have the necrosis. And so that's how we try to improve the bioavailability. So there is no one answer for all the molecules, but we need to work on these different approaches to see that which one can work in the particular molecule.
Thank you, sir.
Yeah, welcome. Is there any other question?
Good afternoon, sir.
Yeah, good afternoon.
Sir, I have a question. Sir, as you mentioned in the previous slides, that
Yes.
that one plant, that Kachnar,
Yes.
so that has anti-inflammatory properties, anti-ulcer properties. But in the case study of 2018, there was mentioned that when it was tested against the gastric ulcers, but instead of curing, it increases the disease in the rat. So like everything was tested properly that it has the property to treat that, but when it was tested, it gets failed. So is there any specific reason why this happened? Do you have any idea?
Nilanshu, right? So, hello.
Yes, sir.
Yeah, actually, your voice was breaking,
Yes, sir.
but I understood one thing. Repeat just to ensure that it is right or wrong. So you said that there was one paper was published in 2018 showing the negative effect or side effect of the particular this particular plant product, right? Gastric ulcer that you were trying to say earlier.
Yes, sir. Yes, sir. Yes, sir.
Yes. Yeah, it is possible. What I would say, this is the problem that most of the time we face, that the reproducibility is not there in this plant products, if I say plant products, which is a general term. Because, the site of collection, the processing, the preparation, all these things. If you don't evaporate the solvent also properly, if you have prepared the extract, let's say ethanolic extract or methanolic extract, whatever the hydroalcoholic extract, if you don't get rid of the thing and the solvents contain some kind of residue thing, unless and until you really make a good analysis of the you process standardized thing, it is difficult to comment on this, and that's the major problem, that the side effects are also there because of this issue. So that's the reason nowadays the concept of phytopharmaceutical has came into the picture, where the CDSCO is approving this. It is not like going through the Ayurveda thing. The CDSCO, which is like a drug controller itself, they are approving now this phytopharmaceutical. And here they are asking for the at least 4 components where that they are there and they are producing the effect. You have identified them properly, and then you are making a formulation of that, and with the proper standardization and everything, you apply for it. You have to perform some efficacy and toxicity study as well and show it. Then only it will go to see the market and all this stuff. So yeah, so I am not surprised to see the negative effect also sometimes, because it is being can be observed if you do not follow the right. In fact, there are if you know that earlier days, all these practitioner, Ayurvedic practitioner, used to have their own gardens, and they used to collect in at the time when they are giving it to the patient. Okay, not like previously, or they knew that when to collect and what to collect and how to collect and all these things. These all things are very important in the plant-based therapies. And so that's the reason, because there are a lot of molecules which might be toxic also in this thing, that you do not want to enrich and give it for this particular effect. I hope I tried to answer these questions in your time.
Yes, yes, sir. Thank you, sir. Thank you.
Okay.
Sir, you talked about the yield of the plant compound.
Yes, yes.
So, sir, there is any method that can be utilized to have a more yield? Like, for example, I can think of like genetic engineering, like using microbes
Yes,
for
yes.
a good production.
Very good point. Very nice point. In fact, that's good you brought this point out. There is a process called as heterologous expression. Okay, so heterologous expression means you are trying to find out the gene which is responsible for the production of these molecules. Okay, and because of the genomic era, you can find out that very quickly nowadays. Now, what the people are doing, they are trying to, from microbial to microbial, or plant to microbes also people are trying. They are trying to get that gene from that particular species, and they create a plasmid, and then they enter into the this particular bacteria or fungi, which as per requirement, or there are some transgenic plants also available, where you are kind of uh, changing the genetic information of that particular plant and trying to improve the yield of your desired molecule. And that is the beauty of this technique. It is not uh, completely, I would say, accepted nowadays because of the various things, but for some of the product it is very well, like for the some antibodies preparation and all, these technologies being used, that's called as plantibodies. The plants are being used to generate this and yeah, there is a shotgun technique. Even some bacteria has also been used for the inducing that particular gene into the particular plant and all this. Yes, so people are using this technologies nowadays to improve the yield, definitely. Yeah, and there is another approach in terms of microbes. If I want to say, there is a OSMAC approach people are following. One strain, uh, OSMAC, many culture condition, where you try to see which is the optimum condition at which you get the maximum yield of the desired product. And so that's the approach. You are screening at a different, like different temperature, different pressure, or different fermentation growth, or fermentation media, pH, all these things you try to optimize, and once it's optimized, use that for the particular process. So yes, it is very important to improve the yield at early stage, because later on it would be difficult, because it is then again you have to change so many things. So at the discovery level itself, try to improve the yield.
Okay, sir. Thank you very much, sir.
Yes, you're welcome. Any other question?
Sir,
Yes.
so while isolating and characterizing compounds from extremophiles, what sort of changes do you make in these techniques, sir? For example, barophytes, what changes do you make in HPLC?
Yeah, a good question. Actually, to be honest with you, we do not mimic, we cannot mimic those conditions here in the lab, to grow these microbes. But we should try to mimic that, because some of the papers that came up with showing that you grow these microbes in those conditions, and then they produce the molecules which are different than at the room temperature or the atmospheric pressure and all. But what we are trying here at this in the NIPER Ahmedabad is that whatever the microbes are growing at our room temperature, means temperature we can increase little bit because we have incubators, but the pressure we keep it atmospheric pressure. And what are the microbial, what are the metabolites they are producing, we are exploring those right now. So mainly we are working on those. And so for HPLC and all, we are trying to see that we get the maximum spread in terms of peaks. And that's how we try to select the stationary phase and solvent system accordingly, like HPLC, mobile phase. Yeah. But we do not have fermenters which can give some pressure and some very high temperature. So like the extreme files grows. So that's right now we do not have that facility. Yeah.
Okay, thank you, sir.
You're welcome. Any other question? No question. So I hope you have got the concept of dereplication. This is not the new word for you now. And I hope you will spread this information also with your colleague, with your friends, because which is very important in the case of natural product research. I hope you agree with me, right?
Yes, sir.
Yes, good. So if no question, we can stop here. I will check with.
Yes, sir. Yes, please.
Yeah.
Sir, thank you so much for your time, and every time
You're
you
welcome.
accept our invitation for this workshop. So it was really informative as usual, and I think there are no more questions. But student, if you have any specific question on this session, or any question on the natural drug discovery, please let me know. I'll convey that to Professor. So,
Yeah, we have time also now. If you have a question, we can further discuss. Nothing related to this topic also, but if you have something related to natural product, if I know the answer, definitely I'll try to answer your question. Yeah.
right. So, students, if you have any question, you are doing any dissertation or project work
Yeah.
related to these things, medicinal plants, anything if you want to ask.
Ma'am, I have one doubt, ma'am.
Okay.
Yes, yes.
Sir, good afternoon, sir. Myself, Nitish Kumar, sir.
Yes. Yes, Nitish.
Sir, I'm doing a project in herbal formulation.
Okay.
I have to make an immune booster tablet, sir.
Okay.
I was okay with all other excipients except hygroscopic content manager, sir.
I didn't get hygroscopic.
So my drug is too hygroscopic, sir.
Okay, okay. Okay.
What type of a herbal excipient to be used, sir?
So your drug is hygroscopic.
Yes, sir.
And you want to, which type of excipients you want to use? So to
Yes, sir.
take care
What
of that
excipients,
particular
sir?
thing.
It should be 100% herbal, sir.
It should be 100% herbal.
Yes, sir. Natural sources.
Okay, I also don't know that by heart right now, but I will also try to find out this because you want to find out basically a excipient which is neutral, which do not give any effect, neither positive nor negative, but at the same time it will make the environment which is like it will absorb the water so that it will not give any effect to the molecule of interest, right? That's what you want looking for.
Yes, sir.
Okay. Yeah, sorry, but I do not know right now immediately answer for this. Yeah.
So somebody
I do
recommended
not know.
to use cornstarch, sir.
Okay.
But it is not effective to the course, sir.
You have tried cornstarch, huh?
Yes, sir. I already tried it, but it is not effective, sir.
Okay, okay. Yeah, because giving answer without knowing it will be difficult. So I need to first look into it properly. Maybe I'll get back to you on this. I'll give it to Dr. Megha and then maybe she can forward that to you. That we can do, definitely.
No problem, sir. So, Nitish, I think you have asked this to Dr. N.P. Yadav from CMAP.
Yes, sir.
Or you can, if you have not asked, then I can give his email ID because he is working specifically on such herbal products and formulation. So
Yes, ma'am.
Yes, yes.
yeah, he can answer this. Or maybe if sir will tell me, then I'll also forward this to you. No problem.
Yes. Because I'm not focusing more on the formulation aspect here, but I know maybe we can find it out. That we will find out then. We'll let you know. Yeah.
Thank you, sir.
No problem.
Hello, sir.
Yes, Akshay.
So I know starch or lactose monohydrate can be used as an excipient.
Right. Okay.
So yeah, maybe Nitish can try that.
He has tried, I think, on starch, he was saying. But yes, lactose monohydrate maybe.
Yes.
Yeah.
So lactose monohydrate may be.
So Nitish, are you hearing this?
Yes, sir.
Yeah. So he's suggesting that you can try lactose monohydrate. Maybe we can also look into it and find out the list, and then we can.
Okay, sir. I will try it and I will report it, sir.
Yeah. Yeah.
Thank you, sir.
Thank you.
So I had another doubt.
Yes.
Saksham this side, sir. So as per the natural things, we because of this antimicrobial resistance, there's a shift in interest towards AMPs as well.
Right. Okay.
So how does the characterization and isolation of AMPs are carried out?
Can you elaborate little bit about AMPs?
Sir, they are basically a small protein molecules, cysteine-rich protein molecules, and they are being known to have antimicrobial properties, anti-cancerous properties, and many such like therapeutic activities. So,
Okay.
like for example, I have been known as theanine is a deficiency theanines are the
Right.
antimicrobial peptides.
Yeah, I
AMP
I
that
came
is
across
adenosine
DKP.
monophosphate.
I came across DKP, diketopiperazine type small 2 amino acid kind of peptide, and there are also like I that is something nowadays the trend is there that the people are more focusing on the proteins and peptides as a therapy. Definitely, it is doing very well, but those particular peptides and proteins I'm not aware about, but diketopiperazines are really doing well. They are also a small 2 amino acid cyclic peptide, and they are showing a very nice activity in these areas. So they are still a lot of things are there in pipelines right now. So that's what they're doing well in there. Yeah.
Okay, sir. Thank you.
Okay.
Okay, so if you have any doubt later, then you can write to me, or you can drop down the message in WhatsApp group, right? So thank you so much, sir. And we always want your support and contribution in such future workshop.
Yes, sure, definitely I'll be there. Thank you so much again.
Thank you, sir.
Thanks. Thank you. Thank you.